Chapter 6 Flowering Plants. Anatomy of flowering plants. You can very easily see the structural similarity and variation in the external morphology of the larger living organism, both plant and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduce you to the internal structure and functional organization of higher plant. Study of internal structure of plant is called anatomy. Plants have cells as the basic unit. Cells are organized into tissue, and in turn the tissue are organized into organ. Different organ in a plant show differences in their internal structure. Within angiosperm, the monocot and dicot are also seen to be anatomically different. Internal structure are also show adaptation to diverse environment. 6.1 The tissue. A tissue is a group of cells having a common origin and usually performing a common function. A plant is made up of different kind of tissue. Tissue are classified into two main groups, namely meristematic and permanent tissue, based on whether the cells being formed are capable of dividing or not. 6.11 Meristematic tissue. Growth in plant is largely restricted to specialized region of active cell division called meristems. That means meristos means divided. Plants have different kinds of meristem. The meristem which occur at the tip of root and shoots and produce primary tissue are called apical meristem. That is given in figure 6.1. Root apical meristem occupy the tip of root, while the shoot apical meristem occupy the distance-most region of the stem axis. During the formation of leaves and elongation of stem and stem, some cells left behind from the shoot apical meristem constitute the axillary bud. Such buds are present in the axil of leaves and are capable of forming a branch or a flower. The meristem which occur between the mature tissue is known as intercalary meristems. They occur in grasses, and regenerates parts are removed by the grazing herbivores. Both apical meristem and intercalary meristems are primary meristems because they appear early in the life of plant and contribute to the formation of the primary plant body. The meristem that occur in the mature region of roots and shoots of many plants, particularly those that produce woody axis and appear later than primary meristem, is called secondary or lateral meristem. They are cylindrical meristem, fascicular, vascular cambium, interfascicular cambium, and core cambium are examples of lateral meristems. These are responsible for producing the secondary tissue. Following division of cell in both primary and as well as secondary meristem, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed permanent or mature cell and constitute the permanent tissue. During the formation of primary plant body, a specific region of the apical meristem produce dermal tissue, ground tissue, and vascular tissue. 6.12 Permanent tissue. The cells of the permanent tissue do not generally divide further. Permanent tissue having all cells similar in structure and function are called simple tissue. Permanent tissue having many different types of cells are called complex tissue. 6.12.1 Simple tissue. A simple tissue is made up of only one type of cells. The various simple tissue in plants are parenchyma, collenchyma, and sclerenchyma. Parenchyma forms the major component within organ. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal, or elongated in shape. Their walls are thin and made up of cellulose. They may either be closely packed or have small intercellular spaces. The parenchyma perform various functions like photosynthesis, storage, and secretion. The collenchyma occur in layers below the epidermis in most of the dicotyledons plant. It is found either as a homogeneous layer or in patches. It consists of cells which are much thickened at the corner due to the deposition of cellulose, hemicellulose, and pectin. Collenchymatous cells may be oval, spherical, or polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intercellular space are absent. They provide mechanical support to the growing parts of the plant such as young stem and petiole of leaf. Sclerenchyma consists of long narrow cells thick and lignified cell wall having a few or numerous pits. They are usually dead and without protoplasts. On the basis of variation in form, structure, origin, and development, sclerenchyma may be either fibrous or sclerites. The fibers are generally thick-walled, elongated, and pointed cells, generally occurring in groups. In various parts of the plant, the sclerites are spherical, oval, or cylindrical, highly thickened dead cells with very narrow cavity. Cavity also means lumen means inside space. These are commonly found in the fruit wall of nuts, pulp of fruit like guava, pear, and sapota, seed coat of legume, leaves of tea. Sclerenchyma provide mechanical support to organ. 6.1 complex tissue. The complex tissue are made of more than one type of cell, and these work together as a unit. Xylem and phloem constitute the complex tissue in plant, that is given in figure 6.3. Xylem function as a conducting tissue for water and minerals from roots to the stem and leaf. It also provides mechanical strength to plant parts. It is composed of four different kind of element, namely tracheids, vessels, xylem fiber, and xylem parenchyma. Gymnosperm lack vessels in their xylem. Tracheids are elongated or tube-like cells with thick and lignified walls and tapering ends. These are dead and are without protoplasm. The inner layer of the cell walls have thickening which vary in form. In flowering plant, tracheids and vessels are the main water transporting elements. Vessel is long cylindrical tube-like structure made up of many cells called vessel members, each with lignified walls and a large central cavity. The vessel cells are also devoid of protoplasm. Vessel members are interconnected through perforation in their common wall. The presence of vessels is a characteristic feature of angiosperm. Xylem fiber have highly thickened walls and obliterated central lumens. These may either be septate or aseptate. Xylem parenchyma cells are living and thin-walled, and their cell walls are made up of cellulose. They store food material in the form of starch or fat and other substances like tannins. The radial conduction of water takes place by the parenchymatous cells. Primary xylem is of two types, protoxylem and metaxylem. The first form primary xylem elements are called protoxylem, and the later form primary xylem is called metaxylem. In stems, the protoxylem lies towards the pith and the metaxylem lies towards the periphery of the organ. This type of primary xylem is called endarch. In roots, the protoxylem lies towards the periphery and metaxylem lies towards the center. Such arrangement of primary xylem is called exarch. Phloem transport food material, usually from leaves to other parts of the plant. Phloem in angiosperm is composed of seed tube elements, companion cells, phloem parenchyma, and phloem fiber. Gymnosperm have albuminous cells and sieve cells. They lack sieve tube and companion cell. Sieve tube elements are long tube-like structure arranged longitudinally and are associated with the companion cell. Their end walls are perforated in a sieve-like manner to form the sieve plates. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lack a nucleus. The function of sieve tubes are controlled by the nucleus of companion cell. The companion cells are specialized parenchymatous cells which are closely associated with sieve tube elements. The sieve tube elements and companion cells are connected by a pit fields present between their common longitudinal wall. The companion cell help in maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmatal connection exists between the cell. The phloem parenchyma store food material and other substances like resins, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledons. Phloem fiber, that is bast fiber, are made up of sclerenchymatous cells. These are generally absent in the primary phloem but are but are found in the secondary phloem. These are much elongated, unbranched, and have pointed needle-like apexes. The cell wall of phloem fiber is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fiber of jute, flax, and hemp are used commercially. The first formed primary phloem consists of narrow sieve tube and is referred to as protophlloem, and later formed phloem have bigger sieve tubes and is referred to as metaphlloem. 6.2 The tissue system. We are discussing types of tissue based on the types of cell present. Let us now consider the how tissue varying depending on their location in the plant body. Their structure and function would also be dependent on location. On the basis of their structure and location, there are three types of tissue system. These are the epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. 6.21 Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cell, stomata, and the epidermal appendages, the trichomes and hair. The epidermis is outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called the cuticle, which prevent the loss of water. Cuticle is absent in roots. Stomata are structure present in the epidermis of leaf. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cell, which encloses stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer walls of guard cells away from the stomal pore are thin, and inner walls towards the stomatal pore are highly thickened. The guard cells possess chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture guard cell and the surrounding subsidiary cells are together called stomatal apparatus, that is given in figure 6.4. The cell of epidermis bear a number of hairs. The root hair are unicellular elongation of the epidermal cell and help absorb water and minerals from the soil. On the stem, the epidermal hair are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. 6.22 The ground tissue system. All tissue except epidermis and vascular bundle constitute the ground tissue. It consists of simple tissue such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith, and medullary rays in the primary stems and root. In leaves, the ground tissue system consists of thin-walled chloroplast containing cells and is called mesophyll. 6.23 The vascular tissue system. The vascular system consists of complex tissue, the phloem and the xylem. The xylem and phloem together constitute the vascular bundle, that is given in figure 6.5. In dicotyledonous stem, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissue and hence are called open vascular bundle. In the monocotyledonous, the vascular bundle have no cambium present in them. Hence, since they do not form secondary tissue, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoint type of vascular bundle, the xylem and the phloem are jointly situated along the same radius of vascular bundle. Such vascular bundle are common in stem and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of xylem. 6.3 Anatomy of dicotyledonous and monocotyledonous plant. For a better understanding of tissue organization of roots, stem and leaves, It is convenient to study the transverse section of the mature zone of this organ. 6.31 Dicotyledonous root. Look at figure 6.6a. It shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of the cell epiblema protrude in the form of unicellular root hair. The cortex consists of several layers of thin-walled parenchyma cells with intercellular space. The innermost layer of the cortex is endodermis. It comprises a single layer of barrel-shaped cells without any intercellular space. The tangential as well as radial walls of the endodermal cells have a deposition of water-impermeable waxy material sovereign in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral root and vascular cambium during the secondary growth takes place in this cell. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later a cambium ring develops between the xylem and phloem. All tissue on the inner side of the endodermis such as pericycle, vascular bundle and pith constitute the stele. 6.32 Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. Figure that is given in figure 6.6b. It has epidermis, cortex, endodermis, pericycle, vascular bundle and pith. As compared to the dicot root which have favor xylem bundle, there are usually more than 6 polyarch xylem bundles in the monocot root. Pith is the large and well-developed. Monocotyledonous roots do not undergo any secondary growth. 6.33 Dicotyledonous stem. The transverse section of a typical young dicotyledonous stem so that the epidermis is the outermost protective layer of the stem. That is given in figure 6.7a, covered with a thin layer of cuticle. It may be a trichomes and a few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of parenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grain, and the layer is also referred to as the starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundle is characteristic of dicot stem. Each vascular bundle is conjoined, open and with enlarged protoxylem. A large number of rounded parenchymatous cells with large intercellular space which occupy the central portion of the stem constitute the pith. 6.34 Monocotyledonous stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. That is given in figure 6.7b. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundle. 6.35 Dorsiventral dicotyledonous leaf. The vertical section of dorsiventral leaf through the lamina shows 3 main parts, namely epidermis, mesophyll and vascular system. The epidermis, which cover both the upper surface adaxial epidermis and lower abaxial epidermis of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bear more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called the mesophyll. Mesophyll, which possess chloroplast and carry out photosynthesis, is made up of Parenchyma. It has two types of cell, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The round and loose. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extend to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system include vascular bundle, which can be seen in the veins and the midrib. The size of the vascular bundle are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle cell. Look at figure 6.8a and find the position of xylem in the vascular bundle. 6.36 Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows following characteristics differences in an isobilateral leaf. The stomata are present on both of the surface of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma that is given in figure 6.8b. In grasses, certain adaxial epidermal cells along the veins modify themselves into large empty colorless cells. These these are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaf curls inwards to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundle, except in main veins, as in vertical section of the leaf. 6.4 Secondary growth. The growth of the root and stem in length with the help of apical meristem is called the primary growth. Apart from primary growth, most dicotyledonous plants exhibit an increase in girth. These increase is called the secondary growth. The tissue involved in secondary growth are lateral meristem, vascular cambium, and core cambium. 6.41 Vascular cambium. The meristematic layer that is responsible for cutting of vascular tissue, xylem and phloem is called vascular cambium. In the young stem, it is present in patches as single layer between the xylem and phloem. Later, it forms a complete ring. 6.41.1 Formation of cambial ring. In dicot stems, the cells of cambium present between the primary xylem and primary phloem is the intrafascicular cambium. The cells of medullary rays adjoining these intrafascicular cambium become meristematic and form the interfascicular cambium. Thus, a continuous ring of cambium is formed. 6.41.2 Activity of the cambial ring. The cambial ring becomes active and begins to cut off new cell both towards the inner and outer side. The cells cut off towards pith mature into secondary xylem, and the cell cut off towards periphery mature in secondary phloem. The cambium is generally more active on the inner side than on the outer. As a result, the amount of secondary xylem produces more than secondary phloem and soon forms a compact mass. The primary and secondary phloems get gradually crushed due to the continued formation and accumulation of secondary xylem. The primary xylem, however, remains more or less intact in or around the center. At some places, the cambium forms a narrow band on the parenchyma, which passes through the secondary xylem and secondary phloem. In the radial direction, these are secondary medullary rays that is given in figure 6.9. 6.41.3 Spring wood and autumn wood. The activity of cambium is under the control of many physiological and environmental factors. In temperate regions, the climatic conditions are not uniform throughout the year. In the spring season, cambium is very active and produces a large number of xylitary elements having vessels with wider cavities. The wood formed during this season is called spring wood or early wood. In winter, the cambium is less active and forms fewer xylitary elements that have narrow vessels, and this wood is called autumn wood or late wood. The spring wood is lighter in color and has a lower density, whereas the autumn wood is darker and has a higher density. The two kinds of woods that appear as alternate concentric rings constitute an annual ring. Annual ring seen in a cut stem give an estimate of the age of the tree. 6.41.4 Heartwood and sapwood. In old trees, the greater part of secondary xylem is dark brown due to deposition of organic compounds like tannins, resins, oils, gums, aromatic substance, and essential oil in the central or innermost layer of the stem. These substances make it hard, durable, and resistant to the attack of microorganisms and insects. This region comprises dead element with highly lignified wall and is called heartwood. The heartwood does not conduct water, but it gives mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color and is known as sapwood. It is involved in the conduction of water and minerals from root to leaf. 6.42 Cork cambium. As the stem continues to increase in girth due to the activity of vascular cambium, the outer cortical and epidermis layer get broken and need to be replaced to provide new protective cell layer. Hence, sooner or later, another meristematic tissue called cork cambium or phallogen develops, usually in the cortex region. Phallogen is a couple of layers thick. It is made of narrow, thin-walled and nearly rectangular cells. Phallogen cuts off cells on both sides. The outer cells differentiate into cork or phallum, while the inner cell differentiate into secondary cortex or phallodome. The cork is impervious to water due to sovereign deposition in the cell wall. The cells of secondary cortex are parenchymatous. Phallogen, phallum, and phallodome are collectively known as periderm. Due to the activity of cork cambium, pressure builds up on the remaining layers peripheral to phallogen, and ultimately these layers die and slow off. Bark is a non-technical term that refers to all the tissue exterior to the vascular cambium, therefore including secondary phloem. Bark refers to number of tissue, that is periderm and secondary phloem. Bark that is formed early in the season is called early or soft bark. Towards the end of the season, late or hard bark is formed. Name the various kind of cell layer which constitute the bark. At certain regions, the phallogen cuts off closely arranged parenchymatous cells on the outer side instead of cork cell. These parenchymatous cells soon rupture the epidermis, forming a lens-shaped opening called lenticles. Lenticles permit the exchange of gases between the outer atmosphere and the internal tissue of the stem. These occur in most woody trees. 6.40 Secondary growth in roots. In the dicot root, the vascular cambium is completely secondary in origin. It originates from the tissue located just below the phloem bundle, a portion of pericycle tissue above the protoxylem, forming a complete and continuous wavy ring, which later becomes circular, that is given in figure 6.11. Further events are similar to those already described above for a dicotyledon stem. Secondary growth also occur in stem and roots of gymnosperm. However, secondary growth does not occur in monocotyledon. Summary. Anatomically, a plant is made up of different kind of tissue. The plant tissue are broadly classified into meristematic, apical, lateral, and intercalary, and permanent tissue, simple and complex. Assimilation of food and its storage, transportation of water, minerals, and photosynthesis, and mechanical support are the main function of tissue. There are three types of tissue system: epidermal, ground, and vascular. The epidermal tissue system are made up of epidermal cells, stomata, and the epidermal appendages. The ground tissue system forms the main bulk of the plant. It is divided into three zones: cortex, pericycle, and pith. The vascular tissue system is formed by the xylem and phloem on the basis of presence of cambium. Location of xylem and phloem. The vascular bundle are of different types. The vascular bundle forming the conducting tissue form the conducting tissue and translocate water, minerals, and food material. Monocotyledon and dicotyledon plants show marked variation in their internal structure. They differ in type, number, and location of vascular bundle. The secondary growth occur in most of the dicotyledonous root and stems, and it increases the girth of the organ by the activity of vascular cambium and the core cambium. The wood is actually a secondary xylem. There are different types of wood on the basis of their composition and time of production. Thank you friends for watching this, for listening this audio, and please post your comment if you want any kind of improvement. मिलते हैं नेक्स्ट वीडियो में, तब तक के लिए बाय. स्टे ट्यून्ड विद बायोलॉजी क्रिटिक एंड एंजॉय द नॉलेज.